Welcome to the Innovation Showcase EBiSC One Stop Shop, how EBiSC operations support sharing and safeguarding of human-induced pluripotent stem cells. My name is Sabine Müller. I am responsible for EBiSC's IT infrastructure and involved with the project's management. The European Bank for Induced Pluripotent Stem Cells, EBiSC, is a centralized non-for-profit IPS bank providing researchers across academia and industry with access to scalable, cost-efficient, and consistent high-quality tools for new medicines development. In the initial phase, 2014 to 2017, EBiSC has been designed to address the increasing demand for quality-controlled, disease-relevant, research-grade IPS lines, data, and cell services. EBiSC launched a second project phase, starting at March 2019, to become a self-sustainable central IPSC hub, pooling EU resources to structure the IPS landscape in terms of quality, consistency, available data, standards, and licensing agreements. Are you regularly using human-induced pluripotent stem cells or planning your first research project? Have you generated human IPS lines that you want to share for future R&D at industry standard? Then you are invited to listen. Not-for-profit EBiSC Biobank has implemented pharma industry's high-quality standards for biobanking of research-grade human iPSCs. In the next couple of minutes, leading EBiSC scientists will highlight how to identify and order IPS lines of interest in the EBiSC online catalog to support your research. And in addition, researchers wishing to leave a legacy and enable future R&D in the area of stem cell research can learn how they can share their achievements with other scientists by a free-of-charge deposition of their IPS lines in the EBiSC Bank while retaining legal ownership.
Hello, everyone, and welcome to our innovation showcase here at the ISSCR. We are glad to introduce our deposition of IPS lines in EBiSC to you. Today, we will have two speakers here for that part. This is Andreas Kurz from Fraunhofer Institute for Biomedical Engineering, IBMT, and myself, I am Ralf Kettenhofen from the IBMT as well. I will give you a brief overview of what we are talking about today. So first of all, Andreas will give you an overview of the consent review process within EBiSC. Then I will take over and talk about the cell line and data capture and the formalized deposition.
My name is Andreas Kurz. I'm a cell biologist with a background in ethics and also genetics. I am responsible for the data management and also somewhat of the ethics framework management in EBiSC. I will present you information about informed consent review process and the ethics framework in EBiSC. I will just go over the main points why we think ethics and informed consent are very important as a part of quality assessment of pluripotent stem cells. First of all, the informed consent by the donor informs and determines the utility of the cell lines. So what can be actually done with the cell lines with the permission of the donor? Therefore, the content review is an integral part of EBiSC quality control, and it is done in EBiSC using a 2I principle by to not miss any information or to misinterpret perhaps information in the informed consent sheets. This process is a manual process. You can find the information how this process is working and organized in EBiSC on the website displayed here on the slide. You also find modal EBiSC consent information templates and consent template. These are framed around other international templates developed, for example, also by the ISSCR, and contains all necessary information which we would like to see or the donor needs to inform about.
Thank you, Andreas. So I will take over now for the cell line data capture. So for the data cell line capture, it is mandatory to register IPS lines into the Human Pluripotent Stem Cell Registry, hiPSC first. hiPSC will provide a unique identifier for each IPS line, which is tractable in publications. And it's very important for you to know that the ownership of IPS lines has to be clarified before the registration. Furthermore, it should be interesting for you that hiPSC also offers an ethics certificate. The certificate includes that the IPS line was derived from full informed consent of the donor, and no undue inducement has been provided for donation. For the IPS line data capture, you first have to create an hiPSC account on the hiPSC homepage. For that, you have to go to the homepage as indicated here. Then you have to provide mandatory information. General information you have to provide comprises the generator institute, the cell line owner, biosample ID, that EBiSC is the distributor of the cells, and that third party can obtain the cell lines. The donor sex and disease diagnosed have to be mentioned, and ethics information has to be provided. The derivation comprises source cell information if a cell line has been modified, the reprobing method, and the vectors used. Furthermore, culture conditions are asking for the medium that it was used. And characterization is mandatory for pluripotency marker gene expression or protein expression, microbiology, and biology. Genotyping information about the karyotype and STR or fingerprint analysis have to be provided as well. And finally, you have to provide information if a genetic modification has been applied to the IPS line. If yes, you have to mention if this modification was disease-related or non-disease related. Means if you have converted disease phenotype into a normal phenotype or vice versa, or if you, for instance, have introduced a reported gene expression cassette. EBiSC also provides online documents and support. Instructions to file all mandatory files during the hiPSC registration can be downloaded from the link below. If online help is required, you can contact hiPSC via the email form from the website, or if you're logged on, using the Send Support Mail button when you're logged into hiPSC. The formalized deposition. Formalized deposition is actually the signing of the EBiSC Material Deposition Agreement, called EMDA. This is a formal contract between the depositor and EBiSC. It allows EBiSC to store, restock if required, and distribute the IPS lines on behalf of the distributor. Just remember, the ownership remains always with the depositor. After the contract is signed, the IPS line can be shipped to the EBiSC banking entities finally.
Hello there, my name is Professor Zamil Kader. I'm a research professor at the University of Oxford and I run a neuroscience research group. I'm also director of the Oxford Headache Centre, and separately I'm director and co-founder of a new startup company called Oxford STEM Tech Limited, where we make stem cells at scale to power precision medicine. And I first came across EBiSC when I was leading the STEM Bank Consortium, which was an innovative medicines initiative, just like EBiSC, in fact. And the STEM Bank program involved generating stem cell lines from 500 different donors, a whole range of patients from Alzheimer's disease to painful neuropathies. And one of the key things that we were looking for as an outcome from the STEM Bank project was a way to ensure sustainability of our research outputs. And EBiSC was the perfect partner to be able to allow this because they were able to take the stem cell lines that we generated from STEM Bank, the very large number, and ensure that they have become available for the broader research use. And that's use both within the international academic community and also by industry researchers as well. And the STEM Bank lines that we made are really high quality stem cell lines. They've passed all of the important quality control metrics. And really importantly, they're also from patients that I think stem cell disease modeling will really make a huge impact on. And EBiSC have made that available to the broader community, which is fantastic. The team really helped with the deposition process, ensuring that we were able to transfer the stem cell lines appropriately and in a secure way. And they've also ensured that the stem cell lines are released to credible researchers. And so the governance around the use of the stem cell lines, as well as the governance around the data associated with the stem cell lines, are taken care of. I think the use of stem cell lines for disease modeling is going to keep on growing. I think increasingly people are understanding the value of stem cell lines for precision medicine, for trying to de-risk drug targets. And so I'm really excited about that future. And I think we'll always need entities like EBiSC to be able to ensure that the logistics of storage of the growing number of stem cell lines remains feasible and possible, and also to ensure that researchers can access them rapidly for their research needs. So I'd just like to finish by thanking EBiSC for the facilities that they've made available. I've certainly benefited from it in my own research. Thank you.
I am Manal Patel. I am a Senior Scientific Manager at the Wellcome Sanger Institute in Cambridge, UK. My background is in pharmacology and neuroscience, and I have 16 years of human embryonic and iPS cell experience. I graduated with a PhD from UCL, and since then have been involved in several successful iPS projects aimed at establishing large resources of human-induced pluripotent stem cells in the UK, consisting of both normal and diseased cohorts, most of which will have substantial genetic data for each cell line produced. My research interests are in cell biology and iPS generation and modeling. More recently, my work has involved spatial transcriptomics and the Human Cell Atlas project to understand the human body by providing spatial context to the sequencing data. I have been working at the Sanger Institute for over 9 years. As part of a collaboration, we were a key depositor of human-induced pluripotent stem cells for the EBiSC project. We deposited a collection of iPS cells into the repository derived from the Human-Induced Pluripotent Stem Cell Project at the Wellcome Sanger Institute. The partnership between Sanger and the EBiSC Consortium allowed the collection of cell lines, including control lines, as well as diseased lines from monogenic diabetes and Bardet-Biedl syndrome patients. The CGAP group, Cellular Generation and Phenotyping Group, within the Sanger Institute expanded and banked 210 human iPS cell lines derived from the hiPSC project. The cell lines were transferred to EBiSC for distribution to the global scientific community. Each cell line was provided with a certificate of analysis showing links to the associated data, such as stem cell marker expression, as well as sterility and identity information, showing the high quality of the cell lines generated. Running the hiPSC and the EBiSC projects in parallel demonstrated the validity of the sterility testing carried out in both projects, as sterility was confirmed both with the EBiSC banks and the hiPSC banks of cells. More information about the cell lines available from both the hiPSC and the EBiSC projects are available in 2 key publications from both projects published in 2017. The hiPSC paper, Common Genetic Variations Driving Molecular Heterogeneity in Human iPS Cells, was published showing the genetic analysis done for the data and the cell lines in the collection. The EBiSC paper was a key launch point of the repository and highlighted how the cell lines were deposited and how the partnership was set up. EBiSC and other scientific groups contribute to the banks at EBiSC, and this allows researchers from around the world to access high quality iPS cell lines with good availability of data. And this enables global development of stem cell research. The EBiSC project was a great example of the Sanger Institute's fundamental principles of sharing data and resources across the world.
Hi, my name's Alex. I'm the Product and Distribution Manager at ECAC, and I'll be talking today about how to order your EBiSC cells.
I'll be talking to you today
about how to order EBiSC lines both directly from ECAC and via our distribution partners. I'll also be discussing the different documents required when ordering EBiSC lines and the delivery of the products to you. But first of all, let's discuss finding the correct IPSC for your research. If you navigate to cells.ebisc.org/search, you'll access the EBiSC catalogue. Here you can browse all 896 lines currently available within the catalogue. You'll be able to search for a specific cell line, gene of interest, cell type, or disease using the search bar at the top of the page. You can also apply filters using the options on the left-hand side. You can find more information about a cell line by navigating to its catalogue page. Here you'll be able to find some general information about the IPSC, databases where information is held, donor information, carrier type, related IPSCs, the reprogramming method, different culture conditions, and characterizations. You'll also be able to find information about any genetic modifications present. To order directly from ECAC, click the Purchase button at the top of each product's page. This will take you directly to the product entry on the ECAC website. You can enter the quantity required in the box on the right-hand side of the page and simply click Add to Cart. A message will appear reminding you to submit an access and use agreement and cell line information pack for your order. You can download them using the 2 links provided, and I will talk about these further later on. Click to proceed to acknowledge this message and to finalize adding the product to your cart. You can view your cart by clicking the link in the top right-hand corner of the page. You can purchase EBiSC lines directly from ECAC, either by using a credit card or a credit account. For more information on how to order directly from ECAC, please visit culturecollections.org.uk/orderinginfo. For the majority of customers, it is easiest to order one of our 3 main distribution partners: Millipore Sigma, KAC, and Cell Bank Australia. Millipore Sigma provides worldwide distribution of all EBiSC products. There are weekly shipments from ECAC to Millipore Sigma in the United States and to their global distribution hub within the United Kingdom. Millipore Sigma provides expert in-country sales and technical support to all EBiSC customers. EBiSC products can be purchased from sigmaaldrich.com. To order an EBiSC product, simply copy the catalogue number found on the product page and enter it into the search bar on sigmaaldrich.com. Navigate to the product page and simply add the product to your cart. Customers in Japan can purchase EBiSC products via our distribution partner, KAC. KAC offers expert advice and support to customers in Japan, and products can be purchased via saibu.jp/product/saibu/IPS. Simply enter the catalogue number into the search bar, add the item to your cart, and check out. Finally, customers in Australia and New Zealand can purchase EBiSC products via Cell Bank Australia. Please contact Cell Bank Australia directly via their website, cellbankaustralia.com, for more information on ordering. To purchase EBiSC products, 2 documents need to be completed. Firstly, the EBiSC Access and Use Agreement, also called the EAUA, needs to be completed for every organization. A cell line information pack or clip also needs to be completed for every cell line that is to be purchased. The clip will outline third-party obligations and also if there are any specific restrictions on that cell line. The EAUA is a legal document and therefore must be completed by somebody granted legal authority within that organization. A clip can be completed by the end user. Multiple clips can be submitted for one EAUA, and it is therefore advisable to think about which lines you might be using in the future and include them when completing this document, as it will speed up any future purchases. One EAUA can be completed and many clips can be attached to it. EBiSC products are safely stored at our cryo storage facility in Salisbury, United Kingdom. If you have ordered directly from EGAC, your order will be dispatched to you by our team and the cold chain maintained using dry ice. Full tracking for all orders is provided, and we will work with you to ensure that your delivery gets to you as quickly as possible. If you order through a distributor, then we will dispatch the products to them first using liquid nitrogen dry shippers. The cold chain is monitored and maintained throughout the journey, and we track the shipments every step of the way. Onward dispatch to you is handled by each distribution partner using dry ice. For further information on EBiSC, please head to ebisc.org. For information on the catalogue, go to cells.ebisc.org. You can also visit culturecollections.org.uk/products/cell-lines/EBiSC for more information, and contact us via culturecollections.org.uk/contact us.
Hello, my name is Charlie Arbour. I'm a Senior Research Fellow here at the Queen Square Institute of Neurology at University College London. Our lab first interacted with EBiSC and its previously related project STEM Bank via the sharing of patient-derived fibroblasts. When collaborators come to us, this interaction with EBiSC has enabled us to point them towards EBiSC to obtain the same lines that we work with. This means that we don't need to organise the shipments or the MTAs, but it also gives us confidence in the quality of the lines between labs and higher reproducibility in the results between us and our collaborators. What's more, it's really nice to know that there's a backup at EBiSC, so heaven forbid there was a problem with the liquid nitrogen supply here at UCL, there's a backup somewhere out there that we can go back to.
So in the last few minutes, leading scientists from EBiSC gave a brief overview how to deposit IPS lines into EBiSC, how to purchase IPS lines in EBiSC, and you also heard some of the EBiSC customers about their experiences with EBiSC. EBiSC has particularly set up an ethical and legal framework to ensure freedom to operate for researchers, including documents providing all clarity about the cell line's details, research use, and contact details upon purchase. So this is how EBiSC operations support sharing and safeguarding of IPS lines. Thank you for listening, and if you have further questions, please do not hesitate to contact us.